Hello everyone, this video is regarding point mutations and the different effects they have. Point mutations are changes in the bases, and there are two kinds, from purine to purine and pyrimidine to pyrimidine, that's transition, and transversion is from purine to pyrimidine and vice versa. When we translate our coding DNA to messenger RNA, the only difference is we're changing our U's, our T's to U's. Everything else stays the same. Now this codes for our amino acids: pyrimidine, alanine, cysteine. This is our stop. This is where actually it ends, but I will put these names for the sake of our first point mutation. Our first point mutation is actually read-through. Read-through is when a stop codon becomes an amino acid and results in a longer protein. So for example here, if we actually change our A to a G, for example, this is no longer a stop codon. This is now tryptophan. So now we continue reading through. Our second mutation is a silent mutation. That codes for the same amino acids, but it's just a different codon. And we'll look at alanine. For example, here, if we change our U to either a G, an A, or a C, they all code for alanine. Our next mutation is nonsense. That's when it changes a codon so that it becomes a stop codon. For example, here, if we have cysteine, UGC, and we change the C to an A, whoops, this is no longer cysteine. Now it's our stop, and this is where it ends. And the last one is missense. That's when one amino acid changes to a totally different one. We're going to look at the tryptophan, and if we replace the U with a G, that it's no longer tryptophan, but that becomes a glycine. This is all about mutations. Thank you.